Quindi, piacere, colleghi, ladies and gentlemen. I would like to thank, first of all, Dr. Fedber for the kind invitation, as well as all the other organizers of this meeting. And it's a privilege for me to share my experience on the epigenetic aspect in the treatment of male infertility. So as we know, infertility in the United States is about 14%. But if we consider here that there is about 1/3, 30% between unexplained and combined form, then indication for infertility is equally distributed between the female and the male partner. So how we evaluate the male partner of infertile couple, and although we proceed with different type of history, family history, surgical history, social and sexual history, then we perform a general exam. But then the key point is to assess the semen analysis. If the semen analysis is normal, then we focus on the female partner. If it's abnormal, then we proceed with other tests, such as evaluation the hormonal profile of the individual, assess for obstruction, etc. And this, most of the time, is done in collaboration with the reproductive urologist, at least here in the US. And therefore, if this is abnormal, then we try to optimize the particular issue. If these results are normal, then we tweak for the best assisted reproductive technology to address the need of the couple. So this is the semen analysis. There are ancillary tests such as abstinence, volume, and viscosity. We focus on concentration and morphology and motility if present. Otherwise, we assess the variability. We also indicate the presence of other cells. It's important to consider that when we treat infertile men, particularly those with azoospermia, this is a study done in 2001 in Japan, 1,500 men, and also been confirmed in our own observation, that about 12 to 13% of these infertile individuals, particularly severely infertile, have chromosomal abnormality. When these can be autosomal, most importantly, gonosomal, and it can be Klinefelter complete mosaic forms. We also can go at a more minute level of abnormality, which is the Y microdeletion. And indeed, in a set of different studies we performed at Cornell, you can see at the tip of the arrow is where the band is missing. These are sequence tag sites that are missing. The base of the arrow is the band present in a normal individual. We know that the incidence of Y chromosome microdeletion increases with the reduction of the sperm production. Indeed, the azoospermic men can be at 8 or 9%. If we are fortunate, though, that if we retrieve spermatozoa from this individual, we can have a fertilization, whether ejaculated testicle of 53% and 40% in this case, and a clinical pregnancy rate of 36% for both semen sorts. When we speak about clinical pregnancy rate throughout this presentation is the presence of at least one fetal heartbeat. When we look at the individual sperm cell, we can assess the aneuploidy of those cells, whether by FISH or by whole exome amplification. And you can see, although in the 2000, we thought that the incidence of surgical retrieved spermatozoa for chromosomal abnormality was much higher than the ejaculated, we have revisited these and published on PLOS ONE in 2019 that actually this is not the case. Now, it is important to consider that the epigenetic aspect of this abnormality can affect, in consequence, the genetic. If you see this morphological abnormality, the so-called decapitated spermatozoa, where there is a sharp angle between the head and the mid-piece of the sperm, this may affect the integrity of the centrosome, as in green, as you can see in this particular case, where we are one of the few centers in the world that actually can assess those for presence and structure and integrity. And once the centrosome or proximal centriole here in the drawing is responsible of ordaining the segregation of chromosomes. And you can see here the distal centriole support the flagellum, the proximal one center of the sperm, we form the spermaster that attract the female pronucleus. And once you reach juxtaposition of the two pronuclei, you have a division of the diplosome here, and here the first mitotic spindle that will ensure the chromosomal normalcy of the first cell after the first cleavage division. Another important aspect of the spermatozoa is a structural organization of the chromatin. It's supercoiled, very compacted, And this is due to the supercoiling of the DNA around the core of protamine. There are residual histone, about 15–20%, so-called linker region, and actually these areas are those that undergo certain damage, or certain fragmentation. We have different assays to assess this: the sperm chromatin structural assay, a chromatin dispersion test here on the left, and on the right, the tunnel assay. How this technique work, this test work, on the left we have the most sensitive comet assay, that the alkaline form of it can assess both fragmentation, mostly single-stranded, while the neutral form assesses the double-stranded DNA break. This can also be done by the structural chromatin assay, and here on the right below, you can see in orange, they can distinguish between the single and the double-stranded, while the tunnel on top can only assess the total fragmentation of the spermatozoa. We know that the sperm chromatin fragmentation is directly correlated with the paternal age, with advancing paternal age, as in this drawing, in this graph. There is also a correlation of the chromatin fragmentation with the length in days of the abstinence periods of the individual. That's the reason why we suggest our infertile couple for the male partner to produce a sample within 24 hours in terms of abstinence. So this is a study that we carried out together with our reproductive urologist, and it's a topographic assessment of the actual chromatin fragmentation in the ejaculate, and then we go proximal into the vas deferens, and you see that from 38% reduced to 22%, and then when we go to the epididymis, it goes to 15–16%, and in the testicle became normal. Remember, our threshold for tunnel is about 15%. So if we assess the data, we identify a certain patient, a certain group of couple that have a high incidence of sperm chromatin fragmentation. And when you see that the fertilization is 66%, the implantation is only 3%, with a clinical pregnancy rate of only 6%. But if we use surgical spermatozoa, although we do not change the fertilization rate, we increase fourfold the implantation rate, the embryo implantation, and we have a clinical pregnancy at 29% and a delivery of 22%. Now, we presented this paper on Fertility in Israel 2014 on perspective of the sperm chromatin integrity. And our take on this paper was that actually we identify an inverse correlation between the DNA fragmentation and the motility, the progressive motility of the spermatozoa. And this has helped us to use this chamber. This is a microfluidic chamber. Actually, we use in particular this chamber here with a 100 microliter, where we can select spermatozoa, the most motile spermatozoa, assuming that they will be actually the spermatozoa that carry the highest chromatin integrity. And it works in this manner. You load the spermatozoa in the lower chamber, and then you allow the most motile to swim in the upper chamber, and you retrieve those that supposedly are intact in terms of chromatin integrity. So we performed this other study where we assessed 69 men, really, actually couple, but looking at the men semen parameter here, you see that when we look at the sample raw, processed by density gradient of microfluidic, we have a reduction in volume. The concentration also reduced dramatically from 28 million to 8 million microfluidic. The motility go from 33%, improve at 60% with density gradient, but get all the way to 97% with microfluidic, and even the morphology improves somewhat. So if we look at the sperm chromatin fragmentation, you see the raw sample is 22%, the density gradient is 18, but the microfluidic is 1.5%. But then has been claimed that the type of breakage is important for the chromosomal normalcy. So if you have total number of chromosome fragmentation is important up to one point, but if we can identify the double-stranded DNA break, then we can see what is the risk of structural chromosomal abnormality. So therefore, the effect on the aneuploidy of the conceptors. So we perform alkaline comet here, and neutral comet, sorry, I apologize. And the neutral comet is specific of the double-stranded DNA fragmentation. And you can see here in number 4 is the longest tail of the comet that indicate the highest level of fragmentation. And this is the proportion of double-stranded DNA break within the total chromatin fragmentation. You can see only 4% in the raw sample, 3% in the density gradient, and negligible in the microfluidic process spermatozoa. So this is an example of the motility of spermatozoa in the raw sample in a maculous chamber. This after density gradient, and in after microfluidic, you can see it's much cleaner, the sample, and the motility is on complete. We have identified 39 couples where we perform a fresh embryo transfer at day 3. So these are couples that actually have a very slim outcome with after ICSI, and therefore we assess the chromatin fragmentation. We know this high DNA fragmentation. So these 39 couples, and this the demographic, you see the age, maternal age and paternal age in the two processing at the same couple. So you see when we selected spermatozoa by density gradient, we performed 25 cycles with a fertilization of 60%. We got 80% of transfer cycle with about 2 embryo. When you have microfluidic, 48 cycle, fertilization of 73%, slightly higher, and even more embryo replacement cycle with the same number of embryo replaced. When you see the comparison here, that you see that with, by density gradient, this couple had only 5% clinical pregnancy rate, all ended up in pregnancy loss. By microfluidic, the clinical pregnancy rate rose to 53%, considering implantation of 27%, and a delivery rate of 46%, minimizing the pregnancy loss. Then we assess another group of couple where we performed PGTA, and we noticed that there were almost no normal embryo to transfer. So 29 couple here, we can see those treated by density gradient, 42 cycle, and 35 with microfluidic. These are the respective maternal and paternal age. Again, the couple is their own control. You see the 42 cycle here with density gradient, resulting in a 62% fertilization rate, with microfluidic about 80%. When we look at the transfer cycle, 28 only with density gradient, 74 for microfluidic. The average embryo replaced was slightly lower for the microfluidic group. When we look at the percentage of embryo that actually underwent PGTA and they euploid, it was only 24% in the density gradient, became all the way to 49% for the microfluidic, so much improvement, I'd say. So when you look at the result, although we were not able to get any pregnancy from those processed by microfluidic, once we treat them, for those processed by density gradient, when we treat them by microfluidic processing, we were able to get 65% implantation with 73% clinical pregnancy rate and 17% delivery rate. So we also try to look at the genetic and epigenetic profile of the infertile couple. So this is another study done on +1, and here we identify 8 control and 5 infertile couple. So what defines the fertile and infertile is here these are a couple that underwent assisted reproductive technology. They have absolutely unexplained infertility, except the concentration, you can see a motility normal and a slightly abnormal morphology, but the ideal candidate for ICSI. And the 8 here on the left were able to achieve a pregnancy with ICSI, while the infertile, no. So we try to study their genetic and epigenetic profile. So when we look at here, the fertilization rate, 79%, 71% for the infertile, the clinical pregnancy rate stay only in one group, in the control. So we remove the DNA directly from the spermatozoa, and we use here the kit, that's the Qiagen kit, and then we qualify here below the graph, you can see the quality of the DNA. And then we use an Illumina platform and machine. And this is the result. On the left here, you see the first 2 samples are fertile donor. And you can see that the duplication and deletion, the mutation are very minute. And so they are comparable in the fertile group, in the people that were able to achieve a pregnancy. That for those unable to achieve a pregnancy, you see the number of duplication and deletion are much more elevated. And that's kind of curve been proven recently during the lifetime of the individual. But also we assess the epigenetic aspect by sequencing the RNA. And we use also a kit from Qiagen here. Then we use the nanodrop for them, the quantification of the RNA, and then qualification by the qubit here, and you see the two peaks in the graph of the ribosomal RNA. And we were able to identify genes that are specific of the development of the acrosome, the PLK4 for the centrosome, the pole here for the mitochondria of the mid-piece, and Casper and spike 16 for the motion of the flagellum. And actually, we even went on to identify specific gene product within the compartment of the spermatozoa, whether in the acrosomal vesicle or in the centrosome or in the flagellum. And when you compare the gene expression, when you have an imbalanced gene expression of those responsible for the motility of the flagellum, you see that there is a direct correlation with the motility on the left, or if they are related to the acrosomal development, they affect the fertilization of the spermatozoa. So we also look at DNA repair gene, or the expression of those genes. We identify the first two, the APLF here, with DNA repair mechanism. The other gene, like the MOR1 for apoptosis, another gene responsible to repress transposable element so that they inhibit mutation. So that's another example of how the epigenetic aspect can affect the genetic profile of the individual. These 7 genes here are represented in this graph, and you see the proportion of them. And so we noticed that when we compare the DNA repair gene expression with the paternal age, there was an inverse correlation, and that's confirmed the previous finding of the DNA fragmentation. And also there is an inverse correlation of this gene function with the DNA fragmentation incidence. So we wanted to look at something more specific, something much more rare, which is the presence of globosospermia. It's less than 0.1% of the infertile male. And you see here on the left, transmission electron microscopy, a normal sperm. In the middle, a globosospermic. And on the right, here, their mid-piece. Fortunately, they have a normal centrosome. That means they can produce or support the development of euploid conceptuses. When we look at the semen parameter of this individual, although this is a very rare form, in our center, we are able to collect a larger number of those in the world, in one single center. We distinguish between partial and complete form. For those partial, you see here the volume is normal, the concentration appears normal, slightly reduced on the average, the motility is compromised, about 50% of normal, the morphology is seriously compromised in the partial form and complete in those complete form, obviously, as you can expect. We look at the retention of histone. This is the control, a normal individual. You see only one sperm here is present that has histone. And here you see the globosospermia of the large majority of the spermatozoa retain a large amount of histones. And that indicates a problem with the compaction and not just the absence of the acrosome. As you do chromatin fragmentation in this individual, you see the characteristic spheric profile of the sperm head. Then we have here all the bioassays we perform in this individual. We have the PLC zeta responsible for activating the oocyte. You see that it's compromised at 27% in the individual with globosospermia. You see the transmission electron microscopy indicated 98% of complete abnormality, of structural, ultrastructural abnormality. The presence of histone is 46%. The sperm chromatin fragmentation is 18%, and the aneuploid is also increased several fold. This is the PLC zeta assessment. We perform this assay in our laboratory, and this is the equatorial localization of the PLC zeta. In the globosospermic individual, is completely missing. So if we look at the clinical outcome of those with partial globosospermia with ICSI, we're actually able to get a fertilization of 34% because we are able to cherry pick and pinpoint those spermatozoa that actually appear normal, they retain some acrosome. We get an embryo implantation of 16%, clinical pregnancy rate, although very minute number of 66%, and so similar delivery. We perform in this case with the complete globosospermia assisted gamete treatment, which in this particular case, we expose the spermatozoa and then the oocyte injected with the sperm with 2-calcium-4. And this will allow the release of the intracellular calcium storage plus the external calcium, and that's responsible for triggering the activation and resumption of meiosis of these injected oocyte. So if we perform complete globosophermia treatment on this individual with a cystogamy treatment, we see that the fertilization rate is 37% from 7%. We have an implantation of 18% and a clinical pregnancy in 50% of the case. Again, these are very minute number of cases, but the syndrome is very rare and multigenic with familiar incident. So if we look at the gene profile of this individual, we identify three couple that we assess the profile of the gene into the spermatozoa, the male partner. We complete globosospermia. The number one patient has a complete failure of implantation. The CG2 has a pregnancy loss at 8 weeks, and the last patient has a full-term pregnancy. And this is a volcano plot of this individual. If you see the gene underexpressed here in CG1, you see that very well imbalanced comparing to the overexpressed, while in the CG2, there is only a problem with the support, the maintenance of the pregnancy. There was implantation, and CG3 has a complete implantation. You see the number of genes just visually, and you can compare on the right with the heat map. They are very minute. So when we look at the gene expression here, you see that the CG1, a gene mainly involved, the CTC2, is involved in embryo implantation, while the NEK2 is involved in mitotic spindle. These were not able to achieve any implantation in this individual, while the CG2 was able to get implantation but fail embryo development as by the imbalance of the gene CBX2 and TGB3, while the remainder is involved in DNA double-strand repair. And is a CG3 with all the gene normally expressed. Then we know that there is another epigenetic aspect, another problem that we identify in this couple, which is the absence of fertilization even after ICSI. This can be due to a low egg yield in the absence or loss of the soluble oocyte activating factor, or the inability of the oocyte actually to be activated by the spermatozoa. So the options are, there have been attempts of recombinant PLC-zeta, cRNA, mRNA. What we use and what throughout the center in the world have been using calcium ionophore. And this is a paper that we published on fertility and sterility on this aspect. And actually, we recruit couple with a low fertilization with ICSI, less than 10%, and we assess the PLC-zeta. The PLC-zeta is present, then we just tweak the stimulation protocol in order to optimize the maturity of the ooplasm, even if there is extrusion of the polar body. And this is a paper that we presented on RBM Online 2016 about the optimal timing of the stimulation between the ACG and the time of retrieval, the ACG and time of removal of the comulus, and the timing of ICSI. But if the PLC-zeta is absent, in that case, we perform a mouse oocyte activation test in order to confirm this absence. And then we carried out a cystogamy treatment, and finally, we perform genomic assessment. So these are the couple, 114 we identified. These are their respective maternal and paternal age. We have a concentration that is 49 million, 43%, and a morphology slightly lower, is about 3%. 119 cycle, we actually able 74% of M2 oocyte, the fertilization rate only 4%. As you can tell, we got some embryo transfer, but no pregnancies. So when we have the presence of PLC-zeta, as in these cases, in this individual, 52 couple, then we treat them in 52 cycles in the control. We have a fertilization rate of only 2%, no pregnancy. But in 105 cycles, we modify the superovulation protocol. We're able to get a fertilization of 59%, a clinical pregnancy rate of 33%, and 25 deliveries. For those instead, when the PLC-zeta was absent, we confirm a mouse oocyte activation test. And here you see when it's successful, you get after this the injection of human sperm into a mouse oocyte. And you can have activation on the left or absence. In this case, we're all confirmed absence of presence of PLC zeta in these men. So 24 couple, their demographic here for maternal and paternal age. And then you have the 27 cycles as used as a control with a fertilization of 9% and no pregnancies. Following assisted gamete treatment, we have 43 cycles. We're able to actually get 79% mature oocytes, 42% fertilization, and 25 cycles with transfer, the pregnancy rate of 36% and 6 deliveries. Established infertility study in 2020. And you can see for the gene expression of this particular individual with the absence of the PLC zeta, this is the most relevant. PLC zeta 1 is affect the calcium oscillation, so the oocyte activation, as well as embryo development. And then the last three, for example, PIC1, SPATA16, and DPY19L, they are involved in the protein kinase C, the most important elongation of the sperm head and acrosome development. And they were all in balance. And these are the proportion of the gene that are in balance. I mentioned the PLC zeta, the first, and the last three on the right here, the most underexpressed. And this, in spite of the presence of this abnormality, we were able to achieve successful pregnancy and delivery, as in this particular picture. Would like to give an overall result of Cornell in the last 28 years. And you see, we have performed 60,000 cycles of assisted reproductive technologies. And of those, 90% nowadays, close to 99% are ICSI. The IVF is about 10% overall this amount of year. And you see of those 43,000, almost 44,000 ICSI cycles, about 10%, 9% with surgical retrieve, the rest with ejaculated spermatozoa. When we look at the result with standard in vitro insemination, we get 60% fertilization, 40% clinical pregnancy, and 35% delivery rate. With ICSI, we obtain a 75% fertilization, similar clinical pregnancy, and similar deliveries. But the fertilization rate is a little biased with ICSI because the denominator is the number of M2 oocyte. So if we correct for the number of retrieve oocyte, you see that the two techniques overlap perfectly. The difference is that ICSI can be adopted, is able to treat couple that cannot benefit from standard in vitro insemination. When we look at the number of newborn, we have well over 21,000 children born at Cornell since the inception of ICSI. So it is important to consider that for male infertility, the indication can be various. It can be from unexplained cryptorchidism and varicocele. They can all affect spermatogenesis up to one point. But the most important is azoospermia, where we don't identify any spermatozoa in their ejaculate. And the azoospermic is the proportion is 23%. And of those azoospermic, about 60% is due to testicular failure of non-obstructive azoospermia. So fortunately for non-obstructive azoospermia, the histopathology here is about 50% of those cases. They have some foci of spermatogenesis. So we retrieve spermatozoa. But for the remainder half, we have equally distributed between maturation arrest and germ cell aplasia. So it's not possible to identify fully developed spermatozoa. The technique to remove these spermatozoa, to retrieve the spermatozoa, are the classical testicular biopsy, or what we master at Cornell is the microtese, where we actually try to identify the most dilated tubules where there are the residual foci of spermatogenesis. When we perform testicular sampling here, we treat couple with obstructive azoospermia, 301, we retrieve spermatozoa in all of them. For the non-obstructive, on 2,647 biopsy, we identify spermatozoa only about 62% of those cases. And so the question is, how can we predict if there is spermatozoa in the testicular biopsy? And the idea is to try to assess certain genes that are present into the ejaculate of this individual, not in the blood lymphocyte, but we actually assess those into the gene. We assess the gene that are present in the RNA actually that is present in the ejaculate. And the sample here on the left, so we can predict the sample on the left here that has spermatozoa, and those on the right that doesn't. So we identify about 10 non-obstructive exospermic men agreed to participate in the study. The age is 37 years. And you can see we identify 45 genes that were imbalanced, and almost all of them, 43, were severely, significantly underexpressed. So we look at the gene involved. You see on the left in the group with spermatozoa, all the genes control some sort of spermatogenesis, spermatid development. So actually, there is a low production, but there's still production of spermatozoa. While on the right here in white, we can see here the gene actually involved in the development of the spermatozoa. In fact, the MLF1 is involved in cell cycle arrest, and then as ADAD1 for spermatid development, another one for apoptosis and even DNA repair. We identified one gene that was specific of the group without sperm, and this for the work so far, but we're still the study is still ongoing. We identified this MSMB, that is a gene involved in inhibin-like with inhibin-like property. So in spite of this difficulty, once we retrieve spermatozoa, we are able to treat this couple. For the obstructive, with 301 cycle, you see the motility, the concentration is across 2 million. The motility 3%, the morphology is sui generis, so they're all abnormal. The fertilization is 66%, and we are able to grant a pregnancy rate of about 42%. When we look for the non-obstructive, 1,600 cycle, the concentration is less than 1 million with 3% motility overall. We have 48% fertilization, although significantly lower than the obstructive, still clinically remarkable. And the pregnancy rate, although lower, still at 36%, clinical pregnancy rate. So we can conclude that the male infertility is a moving target. So we're learning from it. New genes have been identified as we go along. We know more and more, and we investigate. So the gene function and the related environmental factor are responsible for controlling the reproductive potential of this individual. So we need to investigate more the epigenetic of male infertility. So fortunately, this epigenetic imbalance can be addressed somewhat by assisted reproductive technology, particularly with ICSI. So querying the genome and the epigenome, as well as in the near future proteomic, we will be able to tweak the treatment of infertile men. I would like to thank all my collaborators, particularly my group here in the ICSI and andrology, that helped me to put together this presentation. Thank you for your attention.